In this episode, I am joined by Sapna Sharma. Together, we discuss the place of metabolomics in multi-omics studies and how to make the most of the omic data we already have. We'll also learn how to go beyond associations and towards causal relationships and what that means for the future of medicine. The Metabolomist is the podcast where we listen to the stories whispered by metabolomic data. I am Alice Lemonciel, and this season we will examine the application of metabolomics in the clinics and the place of data interpretation in this field. Hello and welcome to the Metabolomist. Today I'm joined by Sapna Sharma, who is a biostatistician and bioinformatician and group leader at the Technical University of Munich in Germany. Hello and welcome.
Hi, Alice.
For people who don't know you and your work, can you begin by explaining what your group works on and what your specialty is?
Sure. Currently, we are focusing on metabolic syndromes. So what it is that it's a cluster of 5 disease conditions, such as hypertension, low HDL cholesterol, high triglyceride, obesity, and diabetes. Mm-hmm. And my focus is in last two parts, obesity and diabetes. And
with the paper we will discuss today.
Yeah. So I started with the adult population. However, in past 2-3 years, I've been growing. My group was growing, and now I have incorporated collaborations regarding this topic from newborn and adolescent as well. Mm-hmm. And the motivation is that these are the root cause for other complex diseases. So it's better to get hands-on as early as possible.
Yeah, and I guess you see signs already of the disease in very early individuals.
Exactly, yeah.
Do you already find trends of things that evolve over time and that people, that like signs that already build on from adolescence into adulthood and before they develop more complex diseases?
Not from my group at least, but what we have is very nice transition from within adolescent, let's say 7 years old, and then how they develop these diseases or after 7 years. So this data we have, and there we can see what goes wrong.
And I know you work with multiple omics, but for this work you focus primarily on metabolomics, or do you always combine it with other things, or what's your favorite strategy?
My background is more on a data science, and I do integrate other data. If you focus on metabolomics, for instance, then you will associate those metabolites with your clinical variable, whatever like disease of interest you have. That is simple, right? And then it gets boring if you keep on doing it for several projects. So yes, then just to bring more spice, I try to integrate as much omics possible. So I take then genomics into account and combine these two layers of information.
We're going to have really nice examples of this when we discuss the papers. We already talked a bit about your background. Is there anything in addition you want to say?
So like early from my school time, I was always interested in maths. I was good in maths. And even in my class, my teachers remembers me as like a person who is of like teaching maths to the same age group. So that was like an inborn interest. And my mom gave
for both math and teaching, interestingly.
Exactly. I wanted to do engineering and, be this cool, do the cool engineering stuff and all that. And she said, maths will take you a long way, and this is something which is really, will never leave you. And that's how all it started, actually. But one thing was very sure after graduation in maths that I didn't want it to do pure maths. I didn't want it to be in a hypothetical world because that engineering thing was always in my head. So I took a path in applied maths where I wanted to apply simple mathematical equation to solve real-world problem, right? And then there was so much more. There was then computations involved. You learn how to automate things. And then slowly, the career went on from simple equation to the complex.
It's very interesting. And now you focus a lot of your work on metabolomics and work with lots of different omics and data types. So why this focus on metabolomics specifically?
Small molecules.
Not lipids. I think we're going to hear a lot about lipids from you, so we should get our story straight from the beginning. Yeah. But small molecules got you interested first.
Not first. So, no,
okay.
No, not at all. During my PhD, I worked a lot with microarray data. Okay. Mm-hmm. And that was all from E. coli bacteria, for instance. Mm-hmm. And then during that time, NGS data was coming up. People were talking about this high throughput data. So I was like, okay, NGS sounds cool. So I went into doing transcriptomics, proteomics, and never paid attention to these level of information. But in 2016, when I joined Helmholtz doing health science, and I was deputy group leader in the Institute of Epidemiology, there I got access to multi-omics. Like, this is something which is so fascinating. And I think I was extremely lucky to have access to different layers of information from a population,
Yeah.
And that's where I found metabolomics. And that in that job, my first challenge was that, okay, you are the person responsible for cleaning, preparing, and handling metabolomics data. So it was targeted, non-targeted. Mm-hmm. And then since 2016, I'm with it, and it has been with me. So
it's very interesting. It's always interesting to see how people get their focus on something, and also you're not the first one to tell me this. Like, there's this idea of being in the right place at the right time, or with the right people. Also, that like in an environment where you get curious about something with people who are curious with you, and then that helps you to go further. That's also something I think in a lot of people, especially who love their work, and you can hear it in the way you describe it. I can see that you really love what you do, that this is sometimes how some of the details of what we choose happen, also based on who we work with and how. Absolutely. Thank you for this. It's a cool story. You mentioned you started cleaning the metabolomic data, and maybe this will be the answer to my next question. But was there something you found particularly difficult when you started working with metabolomics, especially in comparison with other data types that you were used to?
Absolutely, yeah. So other data have some standards, right? I mean, and metabolomics here, the complexity was not clear to me earlier, right? I have been working with different data, and recently I was watching David Wizard's lecture notes, and he put it so well that genomic is based on 4 bases, A, G, C, T, okay, 4. Mm-hmm. If you go to proteomics, then 20 amino acids, and when you come to metabolomics, hallelujah, it's like, damn, you are in this big bang. Yeah. Literally, I see it like a big bang happening in that little chamber of LCMS, and you are exposed to so many small molecules. But then identifying them, how much is known out of like
only a small fraction? Yeah.
So only like I'm just using a words from experts. Like only 10% of the information is known, and the rest all is unknown. Gary Petty, he gave us such a beautiful example of like, look metabolome as a dark room where targeted metabolomics is an approach where you go inside that dark room with the torch, where you put the torch, you see only that space and rest is still there, but you don't know. So it's like amazing, and it's complex, and yet this is developing,
Yeah. Exactly. This is what I was going to get to, because also metabolomics is in a way relatively young from the technological aspect. So we've both been working in this field for a while. So I guess you've also seen this evolution also in the standardization. I mean, it's not what it was 10 or 15 years ago. There is some progress, but we need more progress, right?
Absolutely. Especially targeted is really in a much better stage, and especially some of the kits which provides you real standards, right? I mean, NIST, for instance, is a great example. These are really good for doing clinical trials where you want a good reproducibility, right? So reproducibility is also a challenge in metabolomics.
I know you work a lot from data that has already been generated from samples, so it would be a different approach. But let's say if you were working from the research question from the top down, and you could choose your cohort and you could choose your samples and how you would measure them, would you have like a special approach that you think is best for a specific type of application, or is it more like you find the best workflow for each set of measurements that you have at your disposal?
A very good question. What I have learned growing so far from PhDs and after PhD, all of these, is that everything boils down to money and the funding that you have, right?
That's very good.
I'm really sorry to say. I mean, if I have a budget of 100,000, how can I dream? And I know I have 3,000 samples. How can I dream of untargeted? Although untargeted give me promising novel markers, right?
Yeah.
So it's a very tough battle to choose. I have reached the stage where I can really say openly that, yes, it's a matter of money. And
of course. And do you see then the statistics and bioinformatics that you apply afterwards also helping to focus or to get more chances to find markers or biomarkers if you don't have this level of novelty in the discovery of the molecule themselves?
Exactly.
Maybe where you can compensate for this then.
Exactly. So this is what I'm happy that I have been in this space, because I can see gaps. And once you see gaps, and I personally think there is enough data. There is enough data, but there is not enough bioinformatics tools to make sense out of it. And that's where I have started taking these baby steps where, you have a metabolomics from one study, you have metabolomics from other studies, what we can learn. What we can learn from Jivas, for instance, is people do it, measure it, and keep it available for people to use. But how many of us are using it? And if you go into that, take that just one. It's just like putting on different glass to see different information.
So you have like special glasses for Jivas.
Different omics.
Yeah, and you need the bioinformatician or the biostatistician that can handle this type of data. Like I worked a lot with transcriptomics and all sorts of omics, but GWAS, I don't have the computational capacity to do it myself if I want to work with that. So that's also a level where you need to work with other people with different expertises, and then you can make the best of it. The second paper we'll discuss is a beautiful example of that, because I stopped counting the number of authors at 50, but it's like one of those papers where you have a huge amount of people, and it starts, I guess, from the collection of the samples to the measurements to the analysis. So it takes a village to make a paper like this, but then you have really cool results out of it. Before we dive into the papers, let's talk about the available data, because it's a thought-provoking idea to say there's enough data out there. So let's focus on the type 2 diabetes, because there's definitely a lot of data about type 2 diabetes. If we talk about more obscure diseases, maybe not, but for diseases that are really common like this one, and where people have really been looking into it for a long time and hoping to find something really useful to detect it, to prevent it, to treat it, what makes you say there's enough data? And where do we find this data also?
From my experience, all these big cohorts are meant to share information. So when you say data, the data does not mean that you need exactly, because there is a lot of things happening, right? There is a data protection happening. There is a GDPR we need to be assured of. And most of my data are patient data. So you work very conservative, secure environment like cloud or servers where you process the data. So what I mean by data is information. And I'm telling you, whoever is measuring the data is not measuring for the sake of keeping it. But the information can be transformed in different language, in different forms. For instance, I give you an example of Cora. Okay. Yeah. So, and most of these bigger studies not only measures the data, but has a portal where you can give your research proposal and say that, hey, look, I have done this study and I would like to replicate it and with this data. So these consortiums, of course, these guys, the consortium is not their hardcore job. They are all scientists. They have their own labs. So what they do is that they form a board of members, and they meet like once in a 6 month, or if it's still developing, then every month, once in a month. And they do consider taking this proposal and give you then access to data. So raw data, getting access to raw data is controversial, I understand. But let's say if I want to know the metabolites, those were associated with BMI, okay, then for me, the metabolites and their effect and their significance, this information is enough for me. I don't need raw data. I can rely on their expertise. So they have transformed data into information, And that information is what I'm talking about is possible. And this is where it's our responsibility also, that we should not demand for more, but rather seek for information.
So you work more from the summary statistics of different parameters you're interested in, and that's enough to get you going with the kind of deep analysis that we're going to discuss.
Absolutely. Yes. Yeah.
Very interesting. From your point of view as a biostatistician, is there something you would recommend maybe to people who collect or prepare this data for you that would be useful, that you would like to see?
Definitely, there can be more than, absolutely. You look at the Jivas. Jivas is the best example, right? I mean, if you talk about type 2 diabetes, there is a type 2 diabetes Jivas portal. If you talk about cardiovascular, there is a cardiovascular data. So like that, metabolites needs to grow and make their own portals where you submit. And I'm sure there must be, but it's still not enough. And it's still in the PMID in a supplementary table form. But recently I came across one paper where, like, you do Jivas and then you do meta-analysis. They have started doing it for metabolomics as well. But imagine that it was the first time in Valencia, I guess I heard, that somebody is comparing metabolites from 2 different studies and doing meta-analysis over the known information.
There are a few papers like this, but it's difficult. Yeah, and in one of our episodes this year with Rachel Kelly, we talked about COMETS also, where they gather metabolomic studies from different sources and then do that work of harmonizing it and then making it available in a similar way to Cora. Then so you put a proposal and then you get access to the data that has already been kind of combined for you, and then you can start your work. This really helps people to get more access to more data.
Absolutely. And here, this is the thing. This is where you can fill the gap. In Jivas, a lot of such things are already happened, right? So in metabolomics, it's yet to be done and yet to bring it at that level. So there is a lot to do here. And it's interesting. And
that's a good point. It's a very interesting point. Thank you. Should we get to the first paper we wanted to discuss today?
Absolutely.
Are you ready?
Yes.
So that paper was published 2023. The paper is titled Metabolic Signatures Elucidate the Effect of Body Mass Index on Type 2 Diabetes, which we would think, we know that BMI has a link to Type 2 diabetes, but what were you trying to prove here that was not shown before?
These 2 diseases does not need introduction, right? I mean, 1 in 10 is obese and 1 in 4 is diabetes that we know. The thing is how you can have one omics and multiple clinical variable or clinical information. So again, like in this case, Cora metabolomics data targeted only 150 metabolites we considered was available. For that, we had diabetic and obesity information. But it was always looked at from one angle, either obesity-wise or diabetic-wise. Then I thought there is a crosstalk happening and where it is happening and how we can link it. So that's where I used Mendelian randomization and brought the genetic drivers, because genetic drivers for these 2 so-called disease condition, BMI is a disease condition and diabetic is a disease, we do have a lot of Jivas available, right? And we do have metabolite associated with those Jivas also for both of them. Bang on. Like then I searched for it and I nowhere found this link that can there be one metabolite or set of metabolites that could link these 2 things? So causal inference is what I took. Then
this takes us to your best quote so far from me that I've heard you say a couple of times, that associations are boring. So with this paper, you're going to explain to us how we can go beyond associations and not just stop there.
Exactly. It's just that, being a scientist, you are passionate. And again, also you get bored,
Of course.
Doing one
and
thing.
I have the same feeling. So I'm always trying to push people to go beyond the simple. I mean, it's a lot of work and complex work, but the simple results of the statistics and to try and understand what's going on. It's the same idea. Sure, I can tell you these metabolites are increased, these metabolites are decreased, but it has no impact on what we do about this disease now. We just know that it's like this, and it might not even be like this in the next studies. Let's go into the detail of some figures, and then we can talk about the overall strategy that you used, because I have one question just about the second figure of the paper. It's a set of volcano plots, and there are basically 2 models we were comparing: a basic model with only a few covariates, and then a full model with like the whole shebang. And then you look at which metabolites associate with BMI and which ones with type 2 diabetes. And just for people who, because this is like the very typical way to start looking at things. So could you explain a little bit where you compare different models? You use also different types of models for type 2 diabetes. That is a kind of yes-no categorical states, like you have it or you don't have it. And then BMI, that is a continuous variable. Can you discuss a bit why show the two different models for both, and what does that tell you for the paper? How does that help your story?
Right. First of all, obesity and diabetes both of these diseases have a lifestyle impact on. So it's heavily lifestyle driven. Basically, it's known that if you do any association, your age related and BMI related variables are always confounded. So, but we know that, and we are lucky that in Cora we do have like physical activity, even your social status. It is known that, if you are more literate, how you could keep your life active, and it has a positive effect on your health
in general. Mm-hmm.
We had this, and once you have these variables, you do adjust for it to get a clearer picture. Like you want type 2 diabetes associated metabolites only, and not that it confounds with something else. So that's the main thing. So statistically, if it is a continuous variable or not, you pick your model, you go for logistic or regression. But in the end of the day, you are looking into the associations. And what is the key message here is that you can see, like if you take a basic model, you will have out of 150, 50 metabolites associated, okay, with BMI, which is not true. If you look into the full model, it reduces to 20. Right? Mm-hmm. So now these 20 are more close to the disease, so to say. So you are doing little bit, cleaning, and you're focusing on this is specific markers and not the side actors.
Yeah. And so the figure is a nice message also to tell people, don't just correct for the usual suspect, but if you have the information, try and dig out more of the weeds, basically. Absolutely. So that you get closer to the beautiful flower you're looking for.
Exactly. Well said.
And what's really interesting is, of course, and not surprisingly, but the metabolites that you end up finding are also usual suspects of diabetes and BMI or obesity. So it's alpha-aminoadipic acid has been discussed in that context many times, certain amino acids, and of course certain lipids as well. Mm-hmm. So I guess when you start your study and this is your first result, I guess this is a good start, huh?
Like exactly. This puts you in a right track. I always take yardsticks. I want to see knowns with unknowns, which I would like to make it known.
Mm-hmm. Yes, this is important, right? That you always double check, like before diving too deep into an analysis, to make sure that you're still in sync with what is usually known about that disease.
Yeah.
So otherwise, you can easily go off track and end up in a little corner somewhere by accident.
Yeah. And then you can say that you found a novel marker, but then it's hard to prove and sell.
Maybe now is a good time to discuss how you handle lipids in your work, because here in this analysis, so every metabolite, whether it's a small molecule or a lipid, is considered as a single feature. But of course, for lipids, it can be useful to work with them at the class level, like to group all the phospholipids together or different subclasses of phospholipids together. From your perspective, what would be a couple of advice that you would give to people who start with this?
Yeah, so what I have now started selling lipid is not lipids, but lipid species.
Yeah.
So when you talk about species, then you have many features of that. So this is the terminology that I grabbed from transcriptomics, So you have a gene species. So it falls under, does the same function. They are classified as the same function, but they are different. They are different in metabolites because of 2 carbons, more 2 carbons, less, and forms it. So what I have started doing is that I run each model for one metabolite separately. I don't merge them, but you can do that. You can go for a sum of them, like you can, or to the ratios, for instance. It has been proven that the ratios are more informative in this case, and
that can help. Yeah,
that can help. So it's like talking about not just lipids, but like say in a species form is what I've found useful. And
do you try both usually now when you have data sets with lipids? Do you try each species individually and in parallel the grouping them together?
I do both. Yeah.
Because this is often something that happens is like we try a lot of things and then we keep what gives something interesting.
Exactly.
Right?
Exactly. Yeah. I don't need to go there if my lipids fragmented that I have on hand is causal or associated, and I'm getting a significance result from the data that I have. But in case not, then it is definitely advisable that we look into the ratios or different features. Right? I then also take totally unsupervised learning approach from the machine learning. And there you can see, you just put your clinical data, these lipids together and run this machine learning techniques, and it will give you a selected feature which are responsible for the prediction of the diseases. So that's also a cool idea to how to pick up features, right?
Yeah, and that also helps maybe to cope with one of the criticisms that is often given to metabolomics, that a lot of the features are correlated. So then you focus on one or a few that will hypothetically represent everything that's below the waterline of the iceberg.
Absolutely, yeah.
We're mentioning now a few times GWAS, so the combination of metabolomics with GWAS that we already discussed last year with Carsten Suhr in an episode where we introduced the concept. But for your own work, what is the benefit of adding metabolomics to GWAS and combining those two? How do they benefit from each other?
I checked your podcast and the people there are like my godfathers. Carsten, for instance, he published Metabolite Association, MGWAS we call it, right? And then that's what even I use it. And basically, Mendelian randomization is a method that we use, okay, to the causality. So it estimates the causality. So it's equation-based method, so to say. But why we use it is one funny aspect to it is that it's comparable to a randomized controlled trial. So which is in a randomized controlled trial, you select subjects randomly. Half of them gets medication, half not, bearing in mind their age, sex is same. And then you see the effect of medication, and you compare these two. So the similar thing is happening. In genetics, the SNPs are randomly allocated, right? The SNPs between two might not be the same, but yet causing the same thing. So genetics is what we regard as a random event, and the SNPs, effect alleles and common alleles, these are the two factors where we can compare. Again, it comes with a set of rules. Just like a randomized controlled trial where you make sure your population is falling under the same age and they are healthy. Similarly here, we make sure that these alleles are coming from our exposure of interest, like diabetic and obesity. Yeah. And a lot of data is available for these two diseases. And what we do, then we go and screen for BMI-specific genetics, markers or drivers, and type 2 diabetics specific. So when I say these two things, I'm running this estimation independently, because when you run one causality, you are doing it in one way. You want to see the change in metabolite causing change in diabetes or obesity. But this is like 2 times you are running this method to draw your inference. Basically, it is like association is just a line that these two are connected. Causality is bringing arrow to your line saying that change in this is happening in this direction.
Yes. You've explained nicely how you reach that level of getting closer to causality. Because often what statisticians tell us is that association is not causation. And yes, so that's like the traditional way of saying associations are boring. But now with this, we can really get access to the causal link and the direction of that arrow.
Exactly. Yeah. And these are well-established methods. There are people from Oxford University, from Broad Institute working extensively on getting these tools ready. And what we are doing is that we are using it in context of our scientific questions.
And this is Mendelian randomization. What's the difference with the Sobel mediation test that you also used in the paper?
Because in Sobel, you don't use genetic information. It's simple that one is the independent variable, another one is dependent variable, and mediator is in the middle, which could be what you want to test, right? Whereas here, it's completely different approach. Here, you are looking into a driver that connects you, a driver that connects me, but does not connect something else like our age and all that. And if you move this driver, will you push me or not? So this is like a test between you and me, and this is completely different mathematics algorithms compared to me.
Sure. Yeah. Thank you for clarifying this, because these are often, especially Mendelian randomization, like these are often terms that we hear now and that we see in publications, and we know what the result it means, but it's always nice to get a bit different perspectives on how it works. If you're not an expert, you can grasp a bit better how you get to that conclusion. Thank you for that. You also went to another extra step afterwards where you did pathway analysis that you combined with other experiments. So can you tell us what was that next step that you took, and also where you get that data from that you worked with the extra experiments in mice and everything?
Right. So this is even more challenging because often in data analysis, and you have your analysis, you can easily get lost in the data. But to find the story, the conclusion, is for me is a biological sense of the data. So information is extracted. That's the first step. Now the second step is like biologically what this finding is telling me.
Yeah.
And there I have some examples published where I am the lead author of the paper, and there I investigate, I do data mining, I go into mouse model. There is a mouse consortium, which is for which the same metabolites are measured for obese, non-obese mouse, and feed information. So I go and explore, and this is a little bit tedious. Yeah, you need to spend time, and there is no easy solution because you are in a human world. But you're pushing yourself to go and search in different, and that's where what I have done, I tell my students, I bring my group together saying that, hey, look, these are our scientific questions. Everybody gets to get engaged in finding solutions. And it's so wonderful that somebody is coming with the gene enrichment analysis. Somebody is coming with the mouse models, and somebody is asking some lab in US saying that, hey, you have high-fat diet, low-fat diet, mouse data, how can I get access? They will say, get registered, get access to the data, and then they can,
That's great.
This is time-consuming, I agree.
It's less time-consuming when you do it together, though. It's very interesting to also put the whole group together to find an original solution to a specific problem.
Yeah, and then it becomes the nature of your group, right? Now my group knows that what we want and how we tell our story. And Alice, I should give you a heads up on this, that you talk a lot about like storytelling in science, and this encourages us and even encouraged me in my group to focus on. In the end of the day, your research boils down to the 10-page manuscript.
Yeah,
and that has to be the story, right? Yeah, there should be a story.
I really liked in this paper, and probably in many of your papers as well, like what would be the typical thing would be you have the associations. Now here is the story of what that could mean from the literature. Then after that you go, no, I'm going to look for causality. I'm going to do the Mendelian randomization, and then I'm not going to stop there. I'm going to do these other experiments that personally I would have never thought of with the mouse models. And it's a really great idea. Of course, a mouse is not a human, so you have all the questions of the translation between the species, but that's always the case. But that still gives you very interesting insights into what could be happening and supporting the idea that these changes are really relevant. So it's a great way to go. And I always say also, you can go in many directions. The point is that when you write that manuscript, you justify why you went there, and you draw the conclusions that can be drawn also without overselling whatever finding. And the combined work of different people together is also a way to enrich the solutions that you find at the end. I think it's a really great strategy because it's difficult to get to that level of complexity in the positive sense, like that level of creativity in the analysis. It's hard to do that by yourself. And as soon as you put people together, you start going in different directions, and then it builds on top of each other. So I think it's really a great approach you take.
Thank you.
I think we've discussed most of the main parts of the paper. I would ask you now, overall, what do you think was the most challenging aspect for this type of study in general?
So this is what I learned in this epidemiology world. Here, things starts with analysis plan. And this analysis plan is when I told you that if you want to have a data, you need to give a scientific proposal. When I'm writing a grant, I'm writing a plan. that, which methods I want to do. And this plan is really important. And creating such plans, the biologists are busy doing study design, okay? But now it's our responsibility of a bioinformatician, biostatistician to develop these plans, which is in timely manner executable. A lot depends on your students, your group, your team. You need to first nourish them so that they grow and grow in the direction where you want to, right? I have been extremely lucky that I had a brilliant team. Also, my own experience being in Helmholtz that has given me that I was involved in this planning procedures, which is something which is really essential. And I would put my students, those who are in the second year of PhD, I will take them to these meetings to start learning how to plan your strategies. So, and then it saves a lot of time, right? Then you're not scratching your head that, where I want to go, where. So you need to have a rough roadmap and then start working on it.
So the most challenging part of the work is what we would say is before the work, but it's actually part of it. It's really the first step, the preparation that is going to determine if you can do this study at all. And also, I think it's also great. It's also something you discuss in the book. It's like that first step is also in the book, that as you plan this, you also save yourself a lot of time down the line. Because let's say if we had access to the data just like this, and you could just take data, you could also sit down at your computer and spend 6 months just looking at data with no purpose. And also wouldn't serve you in the end. So I think there's really I think they do this to ensure that the data is used in a good way and for useful things and in a respectful way of the data as well. Do you deviate a lot then from the plan, or?
I was going to tell you this. You need to also be open-minded. You cannot do science.
Can't predict everything that's going to happen.
For instance, we were working on proteomics data. So we did everything what we planned, right? Okay, we will do association, we will do MR, this, that. BMI-related polygenic risk score published. Now that's where that's a sign of good leader who is self-aware of what is happening in market, brings the information, tells them, hey, look, read this paper. Can we do this? Yes, we can do this. In Cora, we have genetic data. We have those polygenic risk scores available. With great leaders like whom you have already interviewed, all of them are amazing. When you collaborate with them, they help you to grow beyond your own comfortable analysis plan.
Yeah.
Then comes the challenge. Take that challenge. Do it. First, you don't like it.
It's unusual. It's uncomfortable.
Yes.
So that's exactly how you can grow. Yeah.
Exactly. And that was the paper which I was going to submit in maximum 5 impact factor, and now we ended up in Nature Communication. And bang, you learn how to bring impact in your science, by being open-minded.
That's a great example. Thanks. Yeah. And in the end, so is that planning what takes the most time? Because that's also a question I usually ask, like for a paper like this one that we discussed, what took the most time from what you remember? From the conception of the study to getting the paper ready?
Starting quite early is important because when you work with this type of data, even if you name it Cora, you need to give this agreement. So you need to start early.
Yeah.
That's my key thing. You need to start early. So something which I've already started doing, planning, irrespective of how much staff I have, because I want that for my own team. I want it for epidemiology team. So getting approval, that was the most challenging. You don't get data just like that.
Yeah. So there's a lot of data available, but you have to earn access to it.
Exactly. And that is all the procedure where you convince committee, not in a verbal way, but in a written way, and get access to all your tools that you need or all your data, and then make your tools to work, pay your.
That takes several months, I guess, even once the proposal is ready. They don't review it so often, so
exactly,
you can start working on something else in the meantime and then come back to it later.
Absolutely, So start early, starting early, and this is now like being a group leader. Now we know we sit and do project planning, we write POVs, like project for future, POF we call it, projects for future. So this is now our responsibility. I would not blame a PhD student to go and search for a topic. That's the time waste.
But that's also part of the mentoring or the direction that you give to the students, also to show, not just to do it for them, but to show them how it's done, and then they can learn at the same time. So it's part of the education as well.
Absolutely.
You also have to do something.
Yeah. Hands-on is always there. I mean,
okay, so I think we're ready to talk about the second paper. We won't get into as much detail for this one, but there were really some interesting findings from this. That one is titled Discovery of Drug Omics Associations in Type 2 Diabetes with Generative Deep Learning Models. And do you want to explain to us what the paper is about and what is the originality of it and the very interesting insights that it brought?
Yeah. So this is a paper coming from Direct Project. It's a consortium where which took stratified approach, not just case control. It was not population based. It was well planned. That okay, we will have a pre-diabetic group, diabetic group, and it's a like pan-European cohort. So what I have learned is that, especially drug information, to transform that data, like which patient took what drug, because that makes it very tedious that there are generics, there are different names, one is using metformin, somebody could use another one, lipid lowering medication. So dealing with this drug data and harmonizing it for 700 subjects that goes into this study was really challenging and took a lot of time. Meanwhile, we were bioinformatician preparing other omics from this study. So I was responsible for metabolomics, targeted, untargeted. Others were like transcriptomics and microbiome and all that. Like that, we distributed work, and there's someone who was working on drug. This data came quite in the end. But once we had it, a lot of effort went into it. Once this was in a readable format, a group from Zorin Baraks from DTU, they are the pioneer of developing new cutting-edge bioinformatic methods. And they gave this deep learning-based framework, multi-omics variational encoder. So this enabled us to integrate multi-omics with the drug data. The findings are amazing. We use this as now small database that how our finding could be affected by drug. What they observe in this, so they use this method, which is beyond univariate analysis. It's complicated. It's deep learning. When I say deep learning, so they take a neural network approach and make this framework ready. But the key finding from this observation, from this analysis, was that how other omics were seen under a drug influence. And the amazing thing was that there are 700 patients, they were given lipid-lowering medication. Okay, so you would think that molecularly it should reflect similarity, but there was differences, which again brings us to the point that, okay, subjects are responding separately and there is a high demand of precision medicine. So and this puts that stepping stone that, look, we found this. We found discriminancy. We found metformin effect on microbiome. It was also not novel and not known yet that how drug could affect your gut microbiome. This is the next topic that is coming in my group. This was the hardcore finding. Starting from developing right tools to process this data, and then this is the information. And this is what then I also used from the same study. I had metabolomics association with diabetics and pre-diabetics. Of course, pre-diabetics was more lucrative topic. Then if I found a biomarker, and I did this analysis already last year, and this paper came this year. So what I did then, I used their information and see if my markers are not affected by drug, but some of them were. Some of them were.
So now you have signatures of the effects of drugs on multiple omics.
That
you can use and look for.
And then I reported in my paper saying that, okay, out of so many, let's say 7 metabolites, 6 of them are under drug. This one is not. Why not? And all that. Then
and how does it work because people with complex diseases like type 2 diabetes tend to have also cardiac problems and all sorts of things. So they will take multiple medications. So you have the signature of one medication, but what happens if you take heart medication, antidiabetics, and so on?
So here they have taken 20 medication into account at the moment. I mean, I'm not sure if we had that data or we have that data and the group is going in that direction of disease complexity, but they took 20 drugs. Mainly they were consisting of lipid lowering medication and diabetic, and these two have a role in inflammatory, like it reduces the inflammation or, and association with omics.
Yeah. Is it different to work on a project like this with several dozens co-authors? It must be a very different approach when you're in a huge consortium like this one.
This is so true. And there are conflicts and everything. But what I have found, no, seriously, because everyone is king of their own kingdom, right?
So
the way to put, first of all, the way to put your art forward comes from your own expertise, okay? And it is generally a trend that when you go up, then the respect towards work increases and comes to your mind, and you will not be rude like we were when we were bachelors. Like when we were young, we will be hot-blooded, we will respond, or very rough, right?
Yes.
But in this hierarchy, when you get experience, suddenly you have huge respect for everything who is doing. So this respect was really, everyone was senior, and seniors had a junior staff. So you need patience, and you learn a lot from the differences.
Yeah, that's also a great way to get ideas for other analysis, I guess.
Exactly. So I don't take it personally, anything. If there is a conflict on anything, I mean, the only thing is that we all work towards one goal, and we all want to grow and learn and have respect. And this is really a cold water. You're going in an ocean, basically. From river, fish is going to ocean. So,
but when it works out, it really gives beautiful results because you have so many different angles to look at a disease or at a set of data sets that you really see. It's also like things that no one can do by themselves again. So at a different scale, but you need all these expertises together to get to that level of insights and analysis.
Also, I would like to add that medical staff has different language of communication. So this project envisions me that how people say things, and there are different ways of communication you can look into and learn from.
Yeah, absolutely. And maybe this is a good time to ask you, is there any advice you would have for someone who wants to do a similar work to what you do, or advice that you wish you had when you were starting off that would have helped you?
I think science is passion-driven. If you have passion, you become patient. You learn patience.
Yes.
You are motivated. Only advice I would give that, the patience, because it's easy to lose, right? And keep it up and be broad-minded. I have learned this,
Stay with the people who keep that passion alive in you as well.
Yeah.
It really matters.
Yeah. There are ways to do it, but this passion is one thing which I would say that is key, which is driving.
Yeah, I agree. It's very important. I think this takes us to my very last question. You know what it is? What is your favorite metabolite and why?
Oh, at the moment, it's lipid.
Always at the moment, yeah.
Lysopeces.
So why lysophosphatidyl-cholines? I'm never sure how people actually pronounce it in English. It's not my first language, so I was wondering, is it I, is it E? But so, LPCs, what about them?
Yeah. So lately, it's coming up everywhere. It's coming up in a pre-diabetic marker, in my adolescence studies also, so which is telling me that it's not only pre-diabetic or pre-obesity related marker in adult, but also, let's say there is a study in India happening where I'm doing the data analysis and everything, and there I find again this thing. So according to age-wise, also early in your life, you can also see this coming up as a marker for obesity or diabetes.
Yes.
So this is what I've from Ulrich Adamski. He put me into this thinking that you don't need to go global, like see what is happening to then fill the gap. So I think this lysopc has a potential to
and does it
different age range.
And does it come from inflammation then? Because often lysopcs are released from the degradation of membrane lipids when we release the precursor for inflammatory molecules.
Very good question. So there is a recently one paper from, if I'm not wrong, from Gabi's group where they were checking the ratio of lysopcs 15 and 17, and were checking in a group of subjects who were had physical activity challenge, and there they found that lysopcs 15 would coming from microbiome, whereas lysopcs 17 is substract. It formed itself, and until now there is no real known information of its biological evidence.
So it's not that simple.
No. The metabolites change themselves as well, right?
So maybe we have then back our versions of inflammation as a precursor for those complex diseases, including diabetes, and the effect on the microbiome as well. So maybe they meet somewhere there at the level of those lipids.
Perhaps.
I need to keep working on this. And figure out the story behind it.
Exactly. This will keep you next 10 years engaged.
In case you were afraid you'd be bored. Thank you very much. It was really a lovely discussion. Thank you very much for taking part in the podcast.
It's only been my pleasure and motivation. So thank you, Alice. Keep doing it.
Thanks. Thank you for joining us in this discussion. I hope that this episode gave you new insights and ideas on how to plan, conduct, and communicate your own metabolomic projects, and that you're excited for the future clinical applications of metabolomics. If you'd like to continue this journey with us, make sure to register for the Metabolomist email list on the podcast's webpage, themetabolomist.com. If you want to learn more about how data interpretation is done, check out my book on the story principle at biocrates.com/thestoryprinciple. For regular news on metabolomics and data interpretation, you can follow me, Alice Limonciel, on LinkedIn, where I post on metabolites, analysis strategies, data processing tools, and more. And make sure to check out our other podcast episodes on the Metabolomist website.